Oh, hello there. How are you? Today I will be talking to you about DNA replication, transcription, and translation, which is the central dogma of biology. Now, if you look here, we have some topics laid out for the lecture today. We start off with replication, transcription, and then translation. You can see the topics here. Take a good look at them. Okay. So now we'll start off with a semi-conservative model of DNA replication. So essentially, if you remember, DNA is anti-parallel. Okay, so that's 5 prime here, 3 prime here, 3 prime here, and 5 prime here. Okay. Now because of this anti-parallel nature of DNA, how can we replicate DNA if we can only add bases to the 3 prime end? Okay. After adding the RNA primer, of course, on both strands. So this is going to be your leading strand, and this over here is going to be your lagging strand. And I will explain why it is your leading and lagging. So it's called leading because it's continuous. So once you have your RNA primer set down here, DNA polymerase can just continuously add bases. However, once I set down RNA primer here, I'm still not on the 3' end, so I can only add a few bases and then set down another RNA primer and keep going. And those small fragments are called Okazaki fragments. Okay, so that is essentially what you have. And so semi-conservative, remember, you have one parent strand and one daughter strand. Okay, sounds good. And then now we move on to transcription. Okay, so transcription is basically your process of going from DNA to messenger RNA, which is important for going into translation, which is where you make your protein. So in transcription, we have something called a promoter region, and this is the DNA nucleotide sequence where your RNA polymerase binds to and starts making your messenger RNA. And one common type of promoter in eukaryotes is the Tata box. Okay, so this is a consensus sequence and it's present in most eukaryotes, almost 99% in eukaryotes. After that, you have something called post-transcriptional processing, and this is a very unique and important function to eukaryotes. And the reason why it's important is because we have compartments in eukaryotic cells, so we are allowed to process mRNAs. So this allows for multiple mRNAs from a single gene, which then translates into multiple proteins. So we do not need a gene for every single protein in the body. A few examples of post-transcriptional processing is going to be the addition of the poly A tail and the GCAP. Both of these are important in preserving the stability and the movement of the messenger RNA out of the nucleus. So after we have covered that, now we're going to jump right into the translation. Translation has 3 stages. It has your initiation, elongation, and termination. Okay, but before I jump into those, I want to mention talking about the codons. Remember, codon consists of 3 nucleotides, and each codon will then have an anticodon, which is brought in by a transfer RNA. And this transfer RNA also brings a particular amino acid which matches with the codon. So initiation, basically, it binds to the P site of the ribosome, and then you constantly add amino acids to your growing amino acid chain as the ribosome moves along the messenger RNA. Now after this, one last thing is going to be your post-transcriptional processing. So you're going to have processing, some processing involved like phosphorylation, glycosylation, and the purpose of this is to target proteins for different areas of the cell. And this concludes my video. Thank you for watching.